Welcome to the University of Pittsburgh Health and Explainable AI podcast, a collaborative initiative between the Health and Explainable AI HexAI Research Laboratory and the Department of Health Information Management at the School of Health and Rehabilitation Sciences, and the Computational Pathology and AI Center of Excellence, CPACE, at the University of Pittsburgh School of Medicine. I'm your host, Jordan Goss-Poire, a health and science reporter. In this podcast series, we explore the transformative integration of responsible and explainable artificial intelligence into health informatics, clinical decision-making, and computational medicine. From reshaping diagnostic accuracy to enhancing patient care pathways, we'll highlight how AI is creating new bridges between researchers, clinicians, and healthcare innovators. Led by Ahmad P. Tafti, Houman Rashidi, and Liron Pantanowitz, the HexAI podcast is committed to democratizing knowledge around ethical, explainable, and clinically relevant AI. Through insightful conversations with domain experts, AI practitioners, and students, we'll spotlight the latest breakthroughs, discuss real-world applications, and unpack the challenges and opportunities that lie ahead in responsible AI in healthcare. So, whether you're a student, practitioner, researcher, or policymaker, this is your gateway to the future of AI-powered healthcare. Today, on this podcast episode, we're going to speak with Freddie Nguyen. He's a physician scientist fellow with the Institute for Medical Engineering and Science at MIT. He's also the chief executive officer at 9 Diagnostics. We're going to discuss MIT's Catalyzing Medical Innovation initiative, which Dr. Nguyen directs. And then we'll touch on diagnostics excellence, molecular design and development, and innovations in precision medicine. Welcome, Dr. Nguyen. Thank you so much for joining us. Would you like to introduce yourself and tell us about the different roles that you filled across the precision medicine field?
Well, thanks so much for inviting me to be on this podcast. It's a great pleasure. Briefly about myself, as you mentioned, I'm a physician scientist engineer by training and by background. And, as a physician scientist, being trained in this field has really been always a focus on what can we do that will maximize patient impact, right? And how can we make a true difference in patients? And that happens certainly on the research side in the early stages of, in the academic space. But at some point, this stuff needs to spin out into the real world, into startups, into companies, and to make it to patients on a much larger scale. And so my role over the years at MIT has been in different places in the innovation spectrum. From, working with a group called MIT Hacking Medicine, which does a lot of healthcare hackathons around the globe, and has really sort of, provided that nexus for people who really are sort of thinking about, well, okay, I want to innovate in this space, but how do I even do it? How do I even approach it? And so, hackathons are a great way for being that, initial coming across that energy activation barrier of seeing what that world is like. To now, in my role as the director of the Catalyst Scholars Program in the MIT Catalyst Program, and really there is thinking about how do you even develop biomedical research programs and projects with that intentionality in mind on the other side, that how can we get these things out to patients a lot quicker? And so how do we, really think about the end goal first and then innovate, from the beginning at that stage, as opposed to what I often say is sort of the 2-step forward, 1-step backwards kind of problem of, you incubate something in the silo of your own lab, and then, it's another year or two before you realize, okay, let me go talk to the clinician on this, or let me talk to the patient on this. And then realizing that there are other constructs in mind that really limit that. So, and across the precision medicine field more specifically, that effort has been really over the last 10 years or so, where a lot of technologies I've worked on has been on the imaging and spectroscopy space, nanotechnology space, a lot of medical diagnostic space, but really with the point in mind of, we have a lot of tools at our disposal. We have a lot of therapeutics at our disposal. But yet, if you look at anything, there's not ever anything that works 100% for every patient that comes through the door. And so that must mean that there's still something we're not capturing. And I think, over the years, we've learned that there's the biological context, but then there's also the social environmental context as well that have an impact in terms of defining who people are. I work a lot in the cancer space, and I always tell people, patients aren't just their one cancer type. They have other conditions, they have other parts of their medical history. But then, in terms of their living environments and their jobs and other things, all those things have an impact on their health and wellness that we need to take into consideration as well.
For young people who are wondering how to frame their views on where advances in precision medicine are heading today and why, what insight would you offer?
Precision medicine has been something we've been trying to work on for decades now in terms of how do we make things more tailored to the individual patient. And largely it has been driven by what are the genetic drivers of a particular disease, and it's how precision medicine has been defined. And really where the field is moving more towards is what's been referred to as functional precision medicine. So yes, you may, have that particular genetic mutation that may, increase your risk factor for the development of that condition or that disease, but it's not necessarily what's going to necessarily express essentially, right, in terms of the protein side of things. And so more of the shift is now shifting from genomics into proteomics and metabolomics and lipidomics, and you can think about all the other different types of omics technologies that are now coming to the forefront. But it's now to really measure what is happening with the patient right now at this particular time of their disease course. So that's been one, change in terms of more technologies that do leverage those different pieces of information. The other has been the efforts around, how do we get better at, essentially ex vivo models or testing, what would work for a patient, but outside of the patient as close as possible, right? So there's been a lot of efforts over the last year to try and move away from animal models because they don't fully recapitulate what is happening, in the human environment. But there's still a lot of gaps and challenges along the way, right? There's been increased advances with organoids and organ on a chip and explant types of technologies. But that's still, that gets us closer to, it's going to be that specific patient's biology that we're testing, the new therapeutics against. But still, it can't fully recapitulate the full in vivo environment in terms of the immune system, for example, and what that reaction might be. And I would say the third area has been, and that's been a big focus of our efforts in diagnostics as well, is really thinking more broadly about the contextual information of the patient. So what I was alluding to earlier in terms of that patients aren't just their singular disease. So now it's the intersectionality of their singular disease that we happen to be looking at, for example, their particular cancer type to, their cardiovascular diseases, their other conditions that may have, their medication history they're taking, what they happen to be living or working at, and how do those come into play as well? Because now when you start looking at proteomics and metabolomics, there's a lot of other things that are going to show up in your bloodstream that aren't, again, just that particular disease that you're studying. And so, those have been now new ways to look at it. And then, of course, now if you think about all these, you have multiple data streams, multiple technologies. And so how do you make sense out of all of this, right? And, that's obviously where a lot of the newer advances in the last few years around AI, machine learning, and computation to really help, pick up patterns where, we suspect they were there all along, but we didn't necessarily know exactly what they were looking, what was there or not to be able to really do that pattern recognition, so to speak. But now we're able to leverage that in a few different ways, right? Whether that's better extraction of the raw signals that we're picking up from individual omics types of technologies, to how do you use AI machine learning in terms of, making sense of multimodal, not just multiplexing, but multimodal types of data, right? And then when you start layering on clinical data and contextual data of the patient, because now you're talking about biological information with social environmental type of information, and how does that layer in all together? Actually, even the technology that we use at Nine Diagnostics, if we look at developing the algorithms based on just the molecular readouts from our technology and the clinical contextual data as two separate algorithms, the sensitivities and specificities are nowhere near where they would be once we combine those two pieces of information together. And so, I think in terms of thinking about, the future, it is moving more and more towards that truly personalized medicine for individual patients, because now it's not just looking at, here's their cancer type, but here's their cancer type in a patient who also has these other conditions, who also happens to be living in these conditions or working in these conditions as well. And how do we make sense of that? Very, very, we thought biology itself was complex, but now when you start incorporating all these, it's even more complex, right? I think that's obviously where a lot of the newer AI technologies are helping us with.
Thinking about advances in molecular design and being able to model how different molecules might interact, how far away are we from being able to model the whole complex organic human health system?
I mean, I wish it could be tomorrow, but I do think, it's just like in a lot of these other tools out there right now is the dearth of data that we have, right? And it's not just amount of data or quantity of data, but it's the quality of data that's always, been the crux of these efforts. And so there's definitely a lot of efforts out there in terms of digital twinning out there, whether that's on the clinical side or the biological side. And I think where the hesitation still comes from is, have we captured everything in a comprehensive manner? Because one of the things I always tell about medicine is that in medicine, we take snapshots in time. We have, tests every few months or every, once a year. And somehow that's supposed to tell us about your day-to-day health and wellness and how that's evolving and happening, right? But you also know that there are some molecules that you measure that even within a single day don't have the same level, right? You look at your cortisol levels, right? You wake up in the morning and you're looking at, certain levels. It's not the same, 12 hours later. And so things like that, where we don't have that kind of visibility, now makes us think like, okay, how well can we take this one snapshot in time and extrapolate it across this digital twin, right? So I think the quantity is certainly one, but also the quality of that data is also going to be important, especially in the context of, as everyone know, around AI and machine learning, sort of a garbage in, garbage out type of scenario as well. So I think, we're certainly moving closer. How quickly can we move there? I think is still a big question mark for me. I think it's going to need to come also from thinking about not just how do we do this with existing data, but how do we create new data streams that truly are better encapsulating of that information? And so right now, up until now, a lot of the efforts in the medical field has been, let's measure this molecule, let's measure that molecule, let's measure this particular test to give us, something about this marker. And how do we go from there to, and this is kind of going back to the concept around really functional testing, which is now going towards, how do we say, okay, I have all these dispersed pieces of data, but to basically just say, my inflammation system is, activated or it's on the rise, or that there's decreased disease burden or increased disease burden in terms of the amount of, cancer, for example, that there is in the system. And so it's really moving from here's sort of discrete markers type of information to here's a function of what is happening with a particular person as well, right? So I think that transition also needs to happen, because once we have that, that can get better at thinking about this digital twinning situation as well.
Through your teaching and through MIT's Catalyzing Medical Innovation initiative, for example, how do you approach introducing young people to these complex topics and steer them into getting involved in pursuing studies in AI and precision medicine?
That's a great question. And I think, working with students and especially younger students is always fun, because, they still have that naivete, right, about what the field is like and what could be there. And so I tell people, especially students, the most important, I would say two important things. One is always starting with the problem, and always starting with a problem that's meaningful, that's impactful, that you're passionate about, that you care about. So that's, first and foremost. And then the second is the intellectual curiosity aspect, but it's really from, don't stop asking the question why. Why is this happening the way it is? Why are we doing this as it has been? Why is this not working? Or why is this working the way it is? Because that's really what's going to drive that innovation process. Oftentimes, we'll get some students who come in and, they know very specific things to do, how to write code and how to, do a particular, a project or algorithm. But, going through the mechanisms of things is, I would say, is a smaller part of the overall project, right? It's trying to think about, what is the problem you're trying to solve? Is it a meaningful problem to solve? And that's not just from a impact perspective, but then once you actually get into the project itself, problem solving is at the heart of almost everything that we do. Whether it's why does this code not work the way it is, or why did we had this hypothesis and then we now are looking at the results and it doesn't look anything like what we thought it was going to look like. So how do we explain it? And so constantly asking that why question, I think, is one of the key things, and that even those of us who have been doing research and doing this innovation process for years and years, there's still a ton that we don't know, right? And that's, I would say, is the driver for all this work.
All right. I'd like to switch topics and talk a little bit about Nine Diagnostics, which you co-founded. What are you and your team studying, and what's Nine Diagnostics' mission?
Yeah, so Nine Diagnostics is a relatively young startup company that was founded a little under 2 years ago, spinning out of Memorial Sloan Kettering Cancer Center. So my co-founders are Dan Heller, who's a professor there, and Mi-Jin Kim, who is a professor at Georgia Tech as well. So, and I have worked on earlier versions of this technology during my time, like postdoc at MIT. But what Nine Diagnostics is focused on is how can we better measure treatment response and effectiveness from cancer patients so that we can drive that feedback loop a lot quicker in terms of, is this treatment actually working for that specific patient or not? Is it at the right dosing for that particular patient? Or should we try to pivot altogether to a new therapy together? If you look at most cancer treatments, treatment response rates are between 20% to 75%, depending on the type of treatment you're looking at. Then, there's been tons of therapeutic development. And for us, when you look at those numbers, the first thing that, pops into our minds is that it's probably that the first treatment you're tried on is probably not going to be the one that's going to be curative for you. And so how do we get through that cycle time a lot faster so that, if treatment one doesn't work, let's not spend 3 to 6 months to figure that out. Let's try and spend, a few weeks and move the patient on to the next part of that, the treatment options that is available to them so that they can benefit a lot earlier. And so the technology is a high throughput proteomics metabolomics platform technology that's based on a new nanosensor technology. And it's the paradigm shift that has happened is that, I was referring to this earlier in terms of looking at individual biomarkers to now really moving to what we have as molecular fingerprinting for the disease state that really captures, all of those different elements that I mentioned before. So not just what's happening on a biological level with that particular cancer that we're looking at, but also what else happens to be going on with that particular patient, as well as their social and environmental factors that comes into that algorithm development. And so their goal is really to go after truly personalized medicine for that individual patient, right? So by having, patients take this test before their treatment, during their treatment, and post-treatment, we can, again, much more tailor what is that response rate to that individual as opposed to looking at things on a population health level as well. But in terms of, to your question about, the learnings along the way to share with people, I think, again, it's starting off with a problem that's truly meaningful and impactful. And for us, patient, I always tell our team, we're patient first, science first company. So, and patients and what we can do for them is truly at the center of our motivation and our mission for the company. And so in that respect, when you talk to cancer patients, a lot of them during this process is, they just want to know, is this moving in the right direction? Or am I in this 3 to 6 months sort of like, I don't know, maybe it's working, maybe it's not working, should I be worried? Should I not be worried? So, that uncertainty level, even during that time, even though, it's very different than the, is it going to actually work or not? But patients have really talked about that uncertainty being, the biggest concern that they have at that point. So even if we can give them some directional movement of it's moving in the right direction or the wrong direction, I think is valuable information for them to have. And I think if you had to also think about this is, always in our program, we always talk about, really looking at things from a multi-stakeholder perspective. So we talked a lot about what it means for the patients. What does it mean for a clinician, right? Who obviously wants to see their patients getting better, but who also wants to see better allocation of resources across the healthcare system too, right? So it's not just that, if they can take care of patients, much more quickly and more effectively, they can see more patients, but also means, other resources in the healthcare system can be allocated to even more patients as well, right? If you think about, obviously the payers and the insurance side of things, that's also thinking about more value-based care type of system, again, helps cut down that overall cost for a particular patient and particular condition. And then, for us, we've been also circling back to thinking about, okay, so that's our long-term vision for the company. What are some, intermediate steps that can get us to that point? Because that's a very, it's a decently long trajectory to get there. And so for us, it's been thinking about, well, what else can we add value with the same kind of information, but yet start to really have an impact at an earlier stage of our company development. And so that has been a focus around the drug development process as well. So our ability to now, go into clinical trials and work with biotech and pharma companies and academic medical centers and be able to provide, again, a quicker feedback loop so that we're not spending that extended amount of time to assess whether a drug is actually working or not and moving it forward. So that helps two ways. One is reducing the life cycle of what the length of a clinical trial could be. But then secondly, it always starts getting us some information of how we can do better in terms of patient stratification, or essentially finding the right patients for that right clinical trial, right? Kind of circling back to what I mentioned earlier is how do we get patients to that most effective treatment a lot quicker? And so that's, for us, that all ties back together, but it's still all in the service of what can we do to help patients.
Before we close our short interview, we'd like to ask all of our guests to offer students a research project idea. We also like to ask our guests for their views on explainable AI. Maybe in this case, could I ask you to offer students a research project idea that touches on explainable AI and that also touches on Nine Diagnostics' work?
Oh, I would say in terms of the explainability component, so I mentioned that, we work a lot with trying to provide that feedback loop and going back and forth between what is quote unquote unknown to what is known, and how does that drive into a constant cycle. And so, for us, even on the technology side, like we do pick up molecules that aren't necessarily known, and we do use it to drive some of the algorithm development and some of the decision making. But ultimately, that circles back to, okay, now that we know these are the things involved, what's the underlying biology, and how does the underlying biology now continue to feed that cycle back? But the other part to also think about is that, especially in this day and age, more data isn't necessarily better data. So I think, one way to also think about explainability is, okay, we may see these other signals, but what does it really mean? And is it really pertinent? Is it really pertinent to this particular decision process, right? So in terms of thinking about new research projects, I think for me, it's always hard to answer this question because I feel like I have 100 different ideas that you could go after. But if I have to provide some constructs, is thinking about, once you figure out what's the clinical problem that you want to tackle, it's really thinking about, what are the pieces of data that are at your disposal? How does it all fit together? And how does it all fit together at the right time and at the right place? And so, we talk about a lot of different ways of, for lack of better terms, poking patients with, right? Whether it's imaging scans, whether it's surveying for particular pieces of information and signs and symptoms to, laboratory testing. But do we really need all of those all the time to really get, to the piece of information that we need to drive a certain decision? And so what is that clinical decision that is most important that you want to focus on? And what are truly the key, not just what key pieces of data, but what key piece of data when, and how frequent that piece of, or the cadence of that piece of data? Because, a lot of times, if you think about patients and, accessibility to certain types of testing and technologies, whether that's from a cost perspective or even from a, like, driving to the next major academic medical center, is not necessarily an easy thing. And so when should we have the right pieces of data to drive those decisions, right? So I don't know if that fully aligns with what your question was, but I think that's probably the best advice I would have in that context.
You touched on explainability. Do you have any thoughts on how to approach the subject of things that we can't explain yet?
I think that's where a lot of, well, obviously now with AI machine learning, sort of that reverse engineering process that you can have, right? Understanding what, either is a disease state or a phenotype at the end being the driver, and then using AI machine learning to backtrack and say, well, what was common between all of these people who had this particular outcome, right? Or even if it's looking at imaging scans, what was common about these features that we saw. That's certainly one aspect in terms of like initially tagging what those are. I think trying to then go back and understand like, well, what is causing that particular feature? I think what's hard is that we often want to think of things in a very linear way, right? We want to say that, A causes B, B causes C, C causes D, and so on to get to that end, outcome that we're looking at. But I think where we're not good at is saying that, okay, there's 5 things that actually need to come together to cause C, for example, to happen. But we may know A and B, but we don't know C, E, and D. And so is this new feature you're looking at also C, or is C causing A and that's then causing, the next factor? So I would say certainly annotating is one aspect, but I think the other part is just keeping your mind open enough for that that the complexity of biology is so vast that, what may be explained a certain way today may not be how it's explained, next week or a year from now as well. And so being open to what we think is the current mechanism of action today may not hold true downstream as well. So,
Dr. Nguyen, it's been a pleasure speaking with you. Would you like to leave us with any final takeaways?
I was going to say, well, thanks so much for having me on the podcast. It was a really fun conversation to have. And, I think I'm always open to meeting with people, whether that's people who have great ideas or new ideas in the healthcare innovation space as well. And, I think at the end of the day, figuring out how can we do better for patients, I'm always passionate about talking to, so don't hesitate to reach out about that. And, I think the other part is, I'm a big proponent of, paying it forward, right? So the opportunities that I've personally gotten up until now in this stage, couldn't have gone without, the people before me. And so, happy to also talk to any students or trainees or researchers who are interested in the space, but also who are interested in figuring out how do you take that leap from the academic world into, startups and innovation world as well. So, but thanks so much for having me.
All right. Thanks again for joining us today. From the University of Pittsburgh, thanks everyone for tuning in and for following the series. Join us again on HexAI, where we explore the transformative integration of responsible and explainable artificial intelligence into health informatics, clinical decision-making, and computational medicine.